Morning, Year 12. So today is the dreaded day. It's time to talk about that cycle I showed you. That's right. Are you ready? I'm ready. Okay, before we start talking about the Krebs cycle, it's important that we remember what we've done so far. We've already done glycolysis, splitting the glucose into two. The two final products of that oxidation reaction was pyruvate. We've got two pyruvate molecules ready to go into the mitochondria to be further oxidized to release the rest of the energy in there that we want. We also made two ATP molecules in that process. Net gain of two, remember we made four in total but had to use two up at the start. And we made two NADH molecules, which are now energized molecules that will drive something that's going to happen later at the very end of this whole process. I'm not going to worry about that bit today though. So what happens next is those pyruvate molecules go into the mitochondria. As you guys learnt about by yourselves last lesson, those molecules undergo something called the link reaction, where they're decarboxylated, where a little bit more energy comes from that molecule. It's locked up with a coenzyme and becomes acetyl coenzyme A. This is essentially pyruvate that's been now altered again, a little bit more energy has been recovered from it, and the final product of the conversion is acetyl coenzyme A. That stuff is ready to go into a huge cycle of reactions that are going to essentially completely rip this molecule apart and take out a ton of energy. Remember that this cycle, just like the link reaction, is going to happen twice for every glucose that we started with, because we broke glucose into two pyruvates. Both of those pyruvates went through their own link reaction. Both of them ended up being converted into acetyl coenzyme A, and both of those acetyl coenzyme A molecules are now going to go into their own Krebs cycle. So everything we're about to talk about happens twice. That means every number we mention now actually needs to be doubled. Okay, so this is the diagram in full. This is about as complex a version of this diagram you'll ever see, and you don't, thankfully, need to know every little bit of this diagram. I like to show it to you just to scare you a bit. What you do need to do is be able to explain the diagram and talk about kind of the main things that happen throughout this cycle, some of the big conversions that happen, why they're happening, and what they're achieving. So we're gonna, instead of looking at this diagram, look at little sections of it one by one until we can build up the full picture, and we're going to talk about what happens at each stage of the cycle. So rather than consider it as a big crazy cycle of loads of chemistry, let's just think of it instead as a series of different chemical reactions. When we put them together, it will end up as a cycle, but each section really is just one chemical reaction. If you can understand those reactions, then you'll be fine. So let's start with the first one. There's one molecule that starts and ends the Krebs cycle, right? It's our starting form, and it's what we will regenerate and create again at the end of the cycle. And that is a molecule called oxaloacetate. Oxaloacetate is a 4-carbon molecule. So usually you'll see it in a diagram just like this one, shown where we have got just 4 circles. They represent the carbon atoms in this molecule. We're not going to show all the detail, that would be ridiculous. And in this reaction you can see that oxaloacetate is reacting with the acetyl coenzyme A molecule. So the 2 carbons that are left in acetyl coenzyme A, remember this was a 3-carbon pyruvate molecule, but one of those carbons was removed, decarboxylated in the link reaction. So it is now a 2-carbon molecule. Those 2 carbons are added to the oxaloacetate, and that builds a molecule called citrate, sometimes called citric acid. Citrate is a 6, therefore, carbon molecule. And that's really just the first step. What we've done is we've fixed the 2 carbons that were in that acetyl coenzyme A into a larger organic molecule. Okay, we've locked them into the oxaloacetate molecule and created citrate. What's going to happen next is we are slowly now going to break those carbons back off, along with the hydrogens that are attached to them, and that is going to release energy and hydrogen ions, which are going to get picked up along the way by our energy carrier molecules. So stage 1, here we go. 1 carbon is removed from citrate, it's decarboxylated. As that carbon comes off, so does some hydrogen. That hydrogen, just like it has been throughout the process so far, is picked up by an NAD+ molecule, which becomes NADH. That now leaves us with a 5-carbon molecule. You don't need to know the names of all these intermediary molecules. I know. Good news, right? Following that, the exact same thing happens a second time. Our 5-carbon molecule is decarboxylated back into a 4-carbon molecule, releasing some hydrogen, releasing some energy, releasing CO2. The hydrogen is picked up by NAD plus again to become NADH again. All these NADH molecules are now energized molecules and they're going to be used later. We're just saving them all up for the final stage of this process that you guys are going to love, by the way. Okay, so once the second decarboxylation has happened, we now have to go through a series of reactions, unfortunately you don't need to know all the details of, where we reorganize that molecule and convert it back into our original molecule of oxaloacetate so that the cycle can go round again. Within that reorganization, more hydrogen gets removed from the molecule. This time it's picked up by 2 hydrogen acceptors. We've got NAD plus again, as we normally have done, but we now encounter a new one, FAD. And FAD, don't get caught up on it, it's just like NAD, different letter, same thing. The only difference is FAD can pick up 2 hydrogens at once, so it becomes FADH2. You'll also notice that a little bit of ATP, a single ATP, is made in this phase. The energy that is released in this part of the process is enough to produce 1 ATP molecule. In other words, to phosphorylate an ADP molecule into ATP. At the end of all this, we end up with exactly what we started with, plus all of these extra energized molecules that we've created along the way. Alright, so now let's just name the different sections of that process overall. To begin with, we have added 2 carbons from our acetyl coenzyme A molecule into an oxaloacetate molecule to create citrate. We've locked 2 carbons into a bigger molecule, and that's called fixation. So the first stage of the cycle is called the carbon fixation stage. After that, we have essentially broken carbons back off the molecule, unleashing all the hydrogen that was in there, breaking all that hydrogen away, and then capturing those rogue hydrogens with our hydrogen acceptors, our NAD plus molecules, and FAD. Then we have taken those carbons back off again, we've ripped them back off, ripping off the hydrogen as we go, and those hydrogens have been accepted by NAD plus and FAD. That means that the original molecule has been oxidized. Hydrogen was removed and the hydrogen was captured by NAD. NAD was reduced in the process. So the second phase of the cycle is called the oxidation phase because we're oxidizing the molecule, removing hydrogen. The last thing we had to do was reorganize the molecule and create our oxaloacetate again so that the cycle is ready to go round again. And that therefore is called the regeneration phase. So those are the 3 phases: carbon fixation, oxidation, and then regeneration. Now if you do the math and think about what's going on here, remember the cycle happens twice for every glucose, or twice for every original glucose molecule. We'll see that we've actually created 2 ATP molecules per molecule of glucose we start with, 6 molecules of NADH, which are going to allow energy storage and transfer later, 2 molecules of FADH2, and we have released 4 molecules of carbon dioxide. That's because the cycles happen twice for every glucose molecule. Now, if you also link in the link reaction here and think about how much CO2 was created in the link reaction, and remember that happens twice too, you'll see that overall 6 CO2 molecules have been produced throughout this entire process so far. 6 CO2, that rings a bell somewhere. So we did glycolysis, split the molecule in 2, We actually made some pyruvate, we made some ATP, we made some NADH. Pyruvate was then moved through the link reaction where it was decarboxylated. We also produced more NADH and that new substance, acetyl coenzyme A. 2 lots of that. Both of those went into their own Krebs cycles, producing more NADH, more ATP, some FADH2, and now we're at the point where we've got everything we need to complete respiration and go into that final stage, which is the electron transport chain. I'm going to save that for next time because it's a bit much for today. Alright, enough for today. Let's do some questions and see how much of this you actually understand.